All right, so we're going to look at photosynthesis. So with photosynthesis, this is our chloroplast. And the chloroplast, we're going to look at two parts, the stroma, which is the liquid part, and then we have the granum or grana. And the granum are stacks of thylakoid membranes. And if we cut open one of those stacks, we can see that the inside has the thylakoid space. Now the chlorophyll is located all within the thylakoid membranes, and that's going to be a part of accepting the light. So what happens first is we're going to look at the light-dependent reaction. And so with the light-dependent reaction, we need light to come down. The light's going to come and shine down on photosystem number 2. This is going to cause photosystem number 2 to just go crazy. And all of a sudden, it's going to excite enough that an electron is going to enter into the electron transport chain. It goes from photosystem 2 and it will enter into a series of proteins where it will be passed around. Because this is now missing an electron, nearby through the process that we call photolysis, water is going to donate an electron to photosystem number 2. We're going to end up with protons and oxygen gas as a waste product. So the protons will be floating around in the thylakoid space. The O2 will be released and will leave the cell when possible. Meanwhile, we have our little electron transport chain. As the electron is passing through, it's going to be drawing more protons from the stroma into the thylakoid space, and these protons are going to play an important role later. The electron will finally make it to a protein we call photosystem number 1, which will also be gathering light energy from the sunlight. And at photosystem number 1, the electron can be used to reduce NADP. The molecule NADP will be here, and that electron, once it is heightened, can reduce that, and we will make NADPH. We refer to this as non-cyclic. The other thing that can happen with that electron, if there is not any NADP around, then the electron can go back and enter back into the electron transport chain and then draw more protons in. So where this is, if this is a limiting reagent and is not available, it will bring more protons. If this is available, then we're going to make the NADPH. We are now building a big proton gradient here, and we're going to enter into a process we call chemiosmosis, which is using the proton gradient of the high concentration inside, the lower concentration outside, and ATP synthase, which is an enzyme that's going to make ATP. So the protons are going to fuel this. So the protons are going to go from greater concentration to lesser concentration through the ATP synthase protein. And as they do that, we're going to combine ADP and inorganic phosphate and make ATP. So this light-dependent reaction requires light to be happening, and it's going to make the products of ATP and NADPH, which are going to be used in the light-independent reaction, and it's going to make oxygen gas, which is a waste product. The best light for this process is blue and red, and the worst light is green. Now we enter into the Calvin cycle, which is also called the light-independent reaction. There are 3 steps to this: the carbon fixation stage, the reduction stage, and the regeneration stage. So the first step, we're going to have CO2, which it's going to get from around the cell, and it's going to join together with a compound RUBP in an enzyme called Rubisco. So Rubisco will take RUBP plus CO2, put them together, and it's going to make RUBP is a 5-carbon molecule, CO2 is a 1-carbon, and in this enzyme it puts them together to make a 6-carbon molecule. Once that happens, that's what we call carbon fixations, because it's adding carbon to it. But that's going to become very unstable, and it's going to break apart into 2 compounds that we call glycerate 3-phosphate. It can also be called 3-phosphoglycerate, and that is going to, these are both 3 carbons, So we had our 6 carbon, which will split into 2 3-carbon molecules. The next part is the reduction. So we're going to use ATP, and we have the NADPH that we had made here is going to be used to reduce our glycerate 3-phosphate, and it's going to make a compound at the end that we call triose phosphate. It also can be called GAP or G3P depending on what textbooks that you are using. That triose phosphate, some of this will come off and it can be used to make glucose. So once triose phosphate is made, the triose phosphate can come down here and go off to make glucose or other carbohydrates, or the triose phosphate can enter and be regenerated back to RUBP. And doing that, it's going to use ATP, and then the triose phosphate will go through a series of steps to go back to becoming RUBP, and then it can fix another carbon, and we can have this cycle. So really, for each carbon dioxide, it will take 6 turns, because we have 6 glucose molecules, for us to make 1 glucose from here. But this triose phosphate can actually go on to make other compounds for the cell, depending on what it needs. Glucose is just one of the most common molecules that it will make at the end of this formation. This process can happen 24 hours a day. It doesn't matter, as long as ATP is around, as long as we have NADPH, and as long as we have CO2, then this can keep happening. These compounds that we got were from the light-dependent reaction, and these are going to happen there. And then that will go off, and the cell can use this glucose for its, it can go off to the mitochondria in the plant cell, or it can be used and stored for other parts of the cell. There we go. Yay!